Chapter 32, Plant Reproduction. As we saw in Chapter 30, there will be a number of terms that will be beyond what you need to know. And so therefore, pay careful attention to what is in the PowerPoint slides and use that to decide the portions of the chapters which are especially needed for you to learn. In addition, in this chapter on plant reproduction, we will focus exclusively on the angiosperms, or the flowering plants, even though the chapter does cover gymnosperms, or the cone-bearing plants. So as we are looking specifically at the flowering plants, or the angiosperms, we will start off with remembering that for all plants there is an alternation of generations. So going from a multicellular gametophyte to a multicellular sporophyte generation. The sporophyte generation is the plant that we see for these flowering plants. And the gametophyte will be located inside of the flower. So if we are talking about a perfect flower that has both female and male components, the female gametophyte will be found in the ovary, and the gametophyte is the ovule, whereas the male gametophyte will be found as part of the stamen, in the anther specifically, and it is the pollen grains. So when we look at flowers in general, they are modified stems bearing modified leaves. So these plants have evolved leaves which help in the pollination process and make them highly successful. Another couple of anatomical parts to keep in mind: the stem of a flower, in the same way we have the stem of a flower. Now, a flower is called a pedicel, where the stem of a leaf is called a petiole. So the pedicel is the flower stem, and then there are a number of circular parts called whorls. The colored whorls around the female and male anatomy are called the petals, and they are usually colored. And the part which initially encases the flower bud and is usually found below the petals are called the sepals. So these are often green whorls, but might be a brownish color as the color may fade, and we found those at the base of the petals. So now let's look up at the parts of the flower. So remember that besides the ovule and the pollen grains, which are the gametophytes, the whole rest of the flower is diploid cells. So they are sporophyte tissue. When we look into the female parts of the flower, altogether they are called the carpel. You may also see them called the pistil, but I usually call them the carpel. The ovary contains ovules, and remember the ovules are the female gametophyte. They are 7 individual cells, so it's a multicellular structure, and we'll take a look more at those details in a little bit. Now, this ovary, which may have one ovule or may have several ovules, is what will become a fruit when the eggs are fertilized. And each ovule becomes a seed. In addition to the ovary, we have a stigma. That's the very top of the carpel. It's usually sticky, and that's where the pollen grains land. And then connecting the stigma to the ovary is a stalk called the style. For the male part of the flower, this is called the stamen, and it's often easy to remember because it has the word men in it. We have the anther, which is the top part, and so this is the structure which contains the pollen grains. Remember the pollen grains, those are the male gametophytes, the multicellular structures that contain sperm and additional support cells. Holding up the anther is the filament. Here is a lab model showing all these parts to a flower. So if we go back through the carpel, here in the center is the carpel, shown in green. And there's usually only one carpel per flower, but there are some flowers that might have multiple carpels. Within each carpel, there is the ovary. Here's the ovary. And then within each ovary, there may be one or more ovules. This model is showing just a single ovule, and you see multiple cells inside, because remember, the ovule is the female gametophyte, which is multicellular. Then on the very top is the stigma, and it's sticky, and you see a couple pollen grains which are stuck to it. And connecting the stigma to the ovary is the style. Then there may be multiple male parts to the flower. So we have a anther. Actually, there's two anthers, one that's opened up or cross-sected, and two anthers on this side. Inside the anther is where we find the pollen grains, which are the male gametophyte. And then supporting each anther is a filament. In this model, we also see colored petals and green sepals, which are located below the petals. So now let's take a closer look at the female gametophyte, the ovule. So this is the multicellular structure, which is inside the ovary. Inside you have a single egg, and then you have an additional 1, 2, 3, 4, 5, 6 cells, for a total of 7 with the egg, 6 cells which are support cells. Now you notice that here one of the cells is this big huge light green structure, and it has 2 nuclei. Then we have 1, 2, 3 cells on the top and 1, 2 cells here on the bottom that actually won't even be used for anything. So the only important cells are the egg itself and this large green cell with the 2 nuclei. And we'll see why that's important in a little bit. Then we have the male gametophyte, which is the pollen grain. So each of these orange circles is one pollen grain. They're located inside the anther. And if we take a closer look at a single pollen grain and we look inside of it, we see that there is a generative cell, and this cell will actually become 2 sperm. Then we see a larger round orange cell with a single nucleus. This is called a tube cell. The tube cell is what's going to make something called the pollen tube to transport the sperm to the egg, because these sperm do not have flagella. So now let's look at the process of pollination. The process of pollination, which is defined as the transfer of pollen grains, from an anther to a stigma. Now, if this happens within the same flower, this is called self-pollination. If it happens between 2 flowers of the same species, then it's called cross-pollination. So self-pollination is within the same flower. Cross-pollination is between 2 flowers, but they must be of the same species. How can this be accomplished? It can be accomplished through wind, insects, birds, or bats, transferring the pollen from the anther to the top of the stigma. So here we see a pollen grain. It has got a generative cell and a tube cell. So the tube cell is going to grow into a pollen tube. So here we see the pollen tube, the one that had the red nucleus, which is growing down to an ovule. Now, if there is more than one ovule, you'll have to have multiple pollen tubes. So each pollen grain can only supply one ovule. How does it know where to grow? It knows where to grow because this flower structure will produce pheromones, which are volatile hormones that will guide the cell on where to grow. The generative cell, which is shown with the blue nucleus, that's going to divide to produce 2 sperm cells. Remember, no flagella on these sperm. So here we had the single blue dividing by meiosis to form 2 sperm cells. Those 2 sperm cells are going to travel down the tube on their way to the ovule. Now, how again does the pollen get to the top of the stigma? Remember, there were 4 basic ways for pollinators. Insects, like bees or butterflies, and the shape of the flower will suit the pollinator. So, for example, flat flowers are better pollinated by moths. So, the flower shape may indicate which kind of pollinator works best for which insects. Now, bats, which are animals, will tend to pollinate flowers that are typically large and bright and visible at night. So sort of bright white flowers that might glow a bit. And they have to be fairly large to fit the head of the bat. Birds. So here birds typically will pollinate flowers that are sturdier so they can support the weight of the bird. But we're talking mostly about hummingbirds, and they're very, very light to begin with. And usually the flowers which are pollinated by birds are bright and not necessarily have to have an odor to them. And lastly, wind. So some flowers come out before the leaves, example maple trees, and they utilize wind to blow the pollen from the anther to the stigma. Now, for all of these pollinators here, these organisms are not pollinating the flowers because of their goodwill. They are actually pollinating the flowers because the flower produces a nectar, which is a sugary syrup that the pollinator is after to consume for food. So while these animals, insects, bats, and birds, are collecting their nectar to eat, then they are, as an after effect, moving pollen from the anthers to the stigma, and it could be within the same flower or it could be across flowers of the same species. So it's a symbiotic kind of relationship and mutualistic in that they both get something out of the deal. Now, the fertilization process, which is the joining of the egg and the sperm, is called a double fertilization. Remember, there were 2 sperm. So what are those 2 sperm doing? Well, here we see our 2 sperm arriving through the pollen tube and about ready to enter into the ovule. So here's a larger picture of the ovule. Remember, there are 7 cells. 1 egg, 1 large light green with 2 nuclei, and 1, 2, 3, 4, 5 cells which will not produce anything. So one of those sperms is going to unite with the egg nucleus. All of these sperm are N. They are haploid. So one is going to unite with the egg, and that will produce the embryo of the plant to come. The other sperm is going to join in with this nucleus that has 2 nuclei to start with. So we have 1, 2, 3 nuclei. So that means it's going to form a triploid. Remember, we have haploid, diploid. This is now triploid, 2 copies of every chromosome. Now that means that it won't produce anything that's alive, but this cell will go on to divide and form many cells that will become what we call the endosperm of the seed. And it is the food that this growing embryo is going to survive on until it is able to germinate and undergo photosynthesis on its own. Here's a YouTube video which you can take a look at, which further explains the process. So when that embryo is formed, when that sperm joins with the egg to form an embryo, then we have now a seed. And seeds are a really important structure for the seeded plants because they allow for a number of important characteristics. Number one, they allow for dormancy under unfavorable conditions. So seeds are generally produced at the end of a growing season, say in the late summer, early fall, and then those seeds have to overwinter possibly some tough conditions, maybe snow or cold weather, and the seed needs to keep the embryo alive until the growing conditions are favorable. Additionally, the seed is going to protect, it has that seed coat, it's going to protect that young plant embryo during the time until it can start to grow. It's going to provide food from the endosperm, and it's going to help disperse the embryos so that they can travel far from their parent plant. We don't necessarily want all of the offspring of a plant to be right next door to it. Remember, plants can't get up and move on their own. So because we have seeds, which are often embedded in fruits that animals eat, then that helps to move the seed with its embryo far away, potentially far away from its parent, and that helps to allow the species to move. Now, during the seed process, when the embryo is inside the seed, the embryo will slow down its metabolic activities. It will essentially stop growing and stop becoming bigger until conditions are right. When the plant does resume growing, that's called germination, and that won't take place until there's enough water. Water is the key factor, and oxygen to reach the embryo. Additionally, temperature can be very, very important, but the most important factor is water. And the seeds are an amazing protective structure. Some seeds from plants have been found from thousands of years ago, and when put into water and given appropriate conditions, they will start to regrow. Some seeds have been found in the tombs of the pharaohs in Egypt and been reintroduced to water, and they've actually sprouted. So I mentioned before that fruits were very, very helpful for dispersing seeds. Keep in mind that a fruit is any plant structure that contains seeds, and they are then officially considered a mature ovary of the carpel. So anything that is a fruit is a mature plant ovary. So during the seed formation, that ovary starts to develop into a fruit. So the other sporophyte tissues around the ovules, because the ovules become the seeds, and they, once they're fertilized, they are also diploid. But all the tissues around the what was the ovule started out being diploid. Now, it is possible for fruits to develop without seed development, and we as human beings have actually bred plants to create fruits, kind of trick them into creating fruits even when the seeds do not develop. So bananas are an example of this, and we can propagate them asexually. So there's no fertilization that actually happens, and so the little dark spots that you see in the bananas are unfertilized ovules. And then for our last part of this chapter, we're going to talk about asexual reproduction. So angiosperms, although they primarily reproduce by sexual reproduction, they can reproduce by asexual reproduction. One way that's done in agriculture quite commonly is called grafting. That's when you take a very valuable plant and you take a cutting of it, you cut a piece of it, and you bind it on to a less valuable stock. So the valuable part is called the scion, and you literally stick it into a rooted stock portion of a less valuable plant. So this is done often in trees. So say you have a very valuable producing apple tree, and you can take lots of grafts or cuttings off of this valuable apple tree, and you can attach it to a less valuable tree, apple tree, and it will just start to regrow and reconnect all those vascular elements. And so you will have the valuable producing part, and you can spread that out through a whole orchard. Additionally, cuttings, and you may have done this even in your own home, that you can take a snip of a leaf or a stem, and you can actually put that into water or plant rooting material and actually create a whole new plant. So remember, in both of these cases, there is no new genetic information in this new plant, that it is an asexual form of reproduction. And we are actually going to take a leaf from an African violet plant, which this is. We're going to take portions of it, and we're going to culture new plants in our lab. And that concludes our material from Chapter 32.